A very good morning to everyone over here. Myself, Dr. Lipi Singh, I'm basically a scientist, stem cell scientist, and working in Advanced Cell Noida, which is a research-based company. And today, especially, I'll thanks the organizing team and chairperson here, who gave me opportunity to talk about the regenerative science and tools, which is maybe helpful for you for treating many female health issues. So today I'm talking more about the stem cells and exosomes. So you may hear more about the stem cells, may not exosomes. So we'll discuss today. So here we go. So I'm starting with the very basic. What are stem cells? So we all know that we have more than 200 kinds of cells. Muscle cells, nerve cells, fat cells. So the origin of these cells are basically from the stem cells, and thus the stem cells are not specialized cells. And when it is differentiated, it can be no longer stem cells. So actually, it is the foundation of all cells, organ, and tissues. So why stem cells are so special? Because it has two key properties. First is self-renewable, and another is plasticity. Self-renew means replicate and become another stem cells. And another is the plasticity means differentiation into another type of cells, like nerve cells, fat cells. So that's the reason it's very special. So what are the types of stem cells? So it depends about this origin. Basically, we put into adult stem cells or embryos. And according to the differentiation process, we call it 4 different terms we use: totipotent stem cell, pluripotent, multipotent, and unipotent. So here you can show it with this. When adult stem, we'll start with the embryos. We have embryonic stem cell, fetal stem cells, and infant, that is water and jelly stem cells. And adult, all the tissue. So we take the terms because very fine difference. So terms, I will more clear in the later slides. What are totipotent? What is a fine difference between all these terms what we used? So you all know about the human embryogenesis. When a sperm and zygote, fertilization is just a sperm and zygote, egg forming a zygote. And during the first 8 weeks of development of fertilization, is very important and complicated process. And you see that in 8 weeks, we are transforming a single cell to the multi-level body plan, multi-cellular organism, where all these systems begin to develop. So now the term is totipotent. Here we call it a zygote, and this stage is totipotent. It can give rise to any types of cells, even the extraembryonic cells. While the pluripotent cells, after 3 days stage when the blastocyst is formed, and there is it can differentiate into 3 germ layers. So these are pluripotent stem cells. But there is some issues of this, ethical issues and teratoma formation. So we are not more in this stem cells. But definitely afterwards, when the pluripotent stem cells differentiate in a specialized lineage, then we call it the multipotent stem cells. So it's more acceptable inside in regenerative science, and also because there is less rejections, less ethical issue, but also difficult to harvest. But it can be overcome with these difficulties. So all we know about the totipotent cells and pluripotent cells is easy to isolate, but because you have a self-renewal, but the problem is here, it's ethical issues and risk of tissue rejection. While multipotent stem cells are more likely less ethical issues, but having limit differentiation. Adult stem cells, what are sources? That is from bone marrow, from fat cells, cord blood, and cord tissue. And we generally deliver, delivery process is intravenous or local delivery. This is the brave history of stem cell research. You can see here, 1963, the first discovery of bone marrow, and then the transplantation between two siblings. Further, there is a identification of the adult stem cells, and further more advancement in induced pluripotent stem cell. We got the Nobel Prize for that. So the basic terms I want to highlight here, because everybody knows the stem cells, but what are the pinpoint you have to remember while using these stem cells or quality purpose? The stem cell markers. So these are actually the specialized protein coating over the cells, which is called receptors, having capability of selecting binding or adhering to signaling molecules. So when these cells bind to the receptor or molecules, they communicate between the cells. So this is very important, and these surface receptors are called stem cell markers, and it's very important to identify the stem cells, and we can sense these stem cells, how many percentage present in those population, we can do by the facts. This is machine available in the diagnostic lab as well. So first stem cells, what we see in the hematopoietic stem cell, that is we found in peripheral blood and the bone marrow and umbilical cord. We all know about that. It is the same property. You see, it can divide in peripheral stem cells and plasticity, that is differentiate into the red blood cell that is specialized. This is called the plasticity of these stem cells. So next is non-hematopoietic stem cell. We call it mesenchymal stem cells, and it is it's a gift for the in the regenerative medicine. You can say, because it has several unique properties it can use easily in clinical aspect. So these are our sources are peripheral blood, adipose, umbilical cord blood, and tissues. It's a property like fibroblast-like structure, you can see here. And there is the tissue, is you can see brown color, is the tissue and the cells coming out from these tissues and spreading and attaching to the cell surface. So, and we do the expansion as well. Other property is, I say, plasticity. That means it can differentiate into other cells like fat cells. This is differentiation of fat cells into fat cells, bone marrow, bone cells, like cell, or cartilage-like cells. So these are the important properties of the mesenchymal stem cell. You have to look before usage of these stem cells. So I talk about the cell markers, stem cell marker. The International Society of Cellular Therapy, this is the international community, we decided what should be the property of the mesenchymal stem cells. Must be plastic adherent in a culture condition, because we have to do in vitro culture expansion in the lab. So you have to these are important things. Another is should express the positive and negative markers. I can explain a little later, or maybe here you can see some of the positive markers there, some of the negative markers, which is present on the MSC markers. So you have to look before use that one. How much percentage there? So it will be given by any you are generating or you are manufacturing, or you can asking for this cell from other company. You have to look for the percentage or report for these markers. Another ICMR guidelines that we have to go for, because we are doing for in vitro culture, I mean the culture system outside the body. So we have to see how many population we can generate. It cannot be unlimited because it's a guideline that 40 population time you have to do, because if you go more in the culture, there is a deletion of, you say, telomerase activity, and cells start aging. So we have to be very particular about these cells. Otherwise, no usage of these cells. And we also use passaging here. Passaging means we have a culture vessel to do the culture. So one culture to another culture with the new media, we call passaging. And it's a guideline for usage of these cells by IMR and the International Society of Cellular Therapy is not more than 4 to 5. So you have to be very particular about these passaging. So why I'm saying the gift to the regenerative medicine, because it has a very specific, characteristic of these stem cells, because there is no required matching by donor and recipient. And so far, so many studies going on, clinical trial going on. So far, no serious side effect has been found and no ethical issues as well. And it has two important properties, immunomodulatory and anti-inflammatory effect of these cells. That is very peculiar about these mesenchymal stem cells. These properties make him more popular in regenerative science. So there is a definite difference between the hematopoietic stem cell and mesenchymal stem cells. And so this very important thing is the matching. For hematopoietic stem cell, you may require matching, but for these mesenchymal, no require matching at all. It's very safe. So why I'm giving importance to the umbilical cord tissue-derived stem cells? Because it's derived from the umbilical cord. And you know about this umbilical cord. It's a connective lifeline between baby and mother. And it has a property of differentiate into other stem cells because plasticity, as you know about the property of the stem cells. So in different cells like neural, adipose, cartilage, bone. So what our lab, that is Advanced Cell, taking lead in producing these stem cells from the Wharton jelly, this area of the cord. So because it's a rich source of mesenchymal, and Wharton jelly, you know that is gelatinous substance in the umbilical cord. This is made up of mucopolysaccharide. Other area also, there we can extract mesenchymal, but the expansion, easily isolation is more from the Wharton jelly. So many clinical trials going on different indications, diabetes, liver, and during COVID also, these mesenchymal stem cells have been used for treatment of this COVID virus. So I told you about the two important properties, that immunomodulatory property, that is the generation of more T regulatory cells. This is very important from these stem cells. Sorry. So another is anti-inflammatory property, and this is a very basic science, so I don't want to go into detail this. So these two important properties make it more popular in regenerative science. So I can go for the video how we produce stem cells in the lab. So exosomes is the part derived from these stem cells. This is cell-free. It's important that stem cell is a cell, but exosome is cell-free, is an extracellular vesicle of particular size. See this video how exosomes form during the culture process of the stem cells. It contains proteins, lipid, messenger RNA, and microRNA. It's very important for cell-to-cell communication. And the particular size of 30 to 150 nanometer is a nanomolecules basically coming out from the cell membrane. And going to the recipient cell where it has to communicate and give the signal what to do next. They have the same property like immunomodulatory, anti-inflammatory, same like stem cells. Important that it is cell-free. So we grow the first stem cells and put it in a culture for some time. They grow the So these are the tangential flow system. We extract the exosome from the stem cells. So here we are doing the tangential filtration process. And we lyophilize because some of the cases we may use for the lyophilized case, some of the currently we put into lyophilization as well and do the quality assurance. We have CD19, 63, and 81. And we do also the protein, how much protein present in that exosome. So we have to see for, and this is the quantification, how much we are giving to the patient for any research use. We manage to and my RNA must be present because without RNA, it's not going to communicate with the other cells. So we have to look for RNA as well. So we do the packaging with that, and the two vials, one is diluent and one is lyophilized exosomes in the pack. So that's all about the exosomes. So these are all the references for we are using for hair restoration. So we have found that exosomes are more better than PRP. Why? Because it has more than growth factors and also mRNA there to communicate with the other cells. So effective more than PRP. And treatment time, because PRP generally we take 3 to 6 times. We have to do that, but it's 1 or 2 is enough. And also, I just want to clear it, the stem cell versus exosomes. I told you that exosomes, stem cells are a little expensive, but it's more affordable, and this contain mRNA. This is very important, and cell-free system, so it's safe and effective. So just I want to take home message for this, because you have to look for those reports what I see before you use, if you go for quality purpose. And these are very important, sterility, CD marking, endotoxin, mycoplasma, and karyotyping, because we do culturing outside the body. There's a telomerase shortening. So this stability must be there. So it is very important before use. I think you have to use, you have given all the tools in regenerative science, so I request you may start with the clinical study or something on these products and see the efficacy. Thank you very much.